Okay, so in the interest of time, I think we will get started. So first off, I want to thank you for attending and welcome to Series 3, or the first webinar in Series 3 of our virtual International Conference on Environmental Ergonomics series. And just for those who are new to it and want to, or else those who want a refresher, it started in April 2020, and the three main organizers have been myself, Stephen Chung from Brock University, Chris Tyler from Roehampton University in the UK, and Tadej Debevic from University of Ljubljana in Slovenia. So we've had two series of 12 talks so far, and they are all available on the Environmental Ergonomics YouTube channel, and they will be hosted there. And if you wish, you can certainly be using them for teaching purposes also. And I know I have, so that I can avoid having to speak all the time, and because of the quality of the speakers. So we have day one today. It's going to be on space physiology, and we have two different speakers and on with different perspectives. Well, hopefully we have two, because the challenge is one of our speakers, Dr. Eric Rivas from Johnson Space Center. He's been trying to get approval for his talks, and as you can imagine, in any big government institution, there are layers upon layers of approval, and Eric is awaiting the final one this morning. So that's why we're going to switch things up a little bit, and we're going to start with Dr. Val Fajardo from Brock University, who is a good colleague of mine and a Canada Research Chair in our Department of Kinesiology. And we will see, hopefully within the next half hour, that Eric will be able to get clearance. And then if so, we will proceed with Eric as our second speaker. If not, we will defer and postpone Eric's talk to another day. So I also encourage you to use the Q&A tool down at the bottom to use that for asking any questions. Type in any questions you want, and then after Val's talk, I will moderate Q&A, and then again, hopefully we'll move on to Eric from there. So with that, I'm going to pass the floor to Dr. Val Fajardo. Again, he's an assistant professor in our Department of Kinesiology and also a Canada Research Chair. And he's going to be talking about muscle unloading and calcium dysregulation and some of the work that he has with mice tissue that have been up to space. So Val, please take it away.
Okay, thank you, Dr. Chung. I do notice that there are two chat notifications. I'm wondering if Dr. Rivas has got that approval, or should I just continue? I don't know if you see that. Oh, okay. I'll just continue. But so thank you for the introduction. And yes, I will be talking about muscle unloading and calcium dysregulation. And specifically, I'm going to be highlighting the work from a PhD student, a really exceptional PhD student in my lab. She actually fast-tracked to the PhD status after 1 year of her master's degree. So she's only in her first term of a PhD. And this is actually, this study characterizing Circa function in murine skeletal muscles after 35 to 37 days of spaceflight. I'm happy to say it was published last week, but it was also just a side project. It's in addition to her dissertation work. And I'm also happy to say that her podcast interview that she had done 2 weeks ago was released yesterday. So for those of you in the audience, I highly recommend that you listen to this podcast. And you'll be seeing a lot of Jessica today throughout my talk. That's just because she's an awesome student, and I actually do think she probably could have given this talk. But anyway, you'll have to deal with me. So what is Circa? Circa is our pet protein in our lab, and it's the Sarcoplasmic Reticulum calcium HbA. So rather than saying that at every mention, I will abbreviate it to Circa. And it's this 10 transmembrane domain protein. So it spans the sarcoplasmic reticulum. So this is the sarcoplasmic reticulum over here on the right. And essentially what this SR is the calcium storage site in all of our muscle cells. So it stores calcium. And you can find Circa right over here on the membrane. And what it does is it catalyzes the active transport of calcium against its concentration gradient into the sarcoplasmic reticulum. So against its concentration gradient, essentially what I want you to think about is maybe pushing something uphill. That's going to require energy. So Circa is trying to push calcium uphill, and that's why we call it active transport. And it does this by using the energy harnessed from ATP hydrolysis. So ATP is that cellular currency of energy. So it uses ATP, the energy from ATP, to bring calcium from the cytosol into the SR. Now I have this point here suggesting that Circa is important for muscle relaxation and ensuring a sufficient calcium load in the SR. So when we want our muscles to contract, we get this electrical signal, an action potential that propagates from the sarcolemmal membrane or the plasma membrane, and it goes down to the transverse tubule. This is the transverse tubule here. And this change in voltage is sensed by these green proteins called the dihydropyridine receptors, which are intimately connected with the ryanodine receptors shown here in orange. And so when dihydropyridine receptors sense this change in voltage, it undergoes this conformational change, and it relays it to the ryanodine receptors, allowing them to open. And that releases the calcium from the storage site, the sarcoplasmic reticulum, and that allows for the activation of the myofilament, so allowing for actin and myosin to bind together to generate force. So that's for the contraction side of things. However, when we want to relax, that calcium signal that activated the myofilaments needs to be taken back into that storage site. And again, that is done by the Circa pumps that will bring calcium back into the SR. So the Circa pumps, yes, are very important for muscle relaxation, but by refilling the sarcoplasmic reticulum, it also ensures there's enough calcium for the next or subsequent muscle contraction. And so with spaceflight, we know that when the astronauts go up in space, or even mice, when they go up in space, their muscles will be unloaded and will experience a certain extent of disuse, particularly the postural muscles. So you'll see a lot of soleus muscle data. So you could see here, when you're standing, the soleus muscle is on, trying to withstand that never-ending downward pull of gravity. And so when the astronauts or mice, they go up in space, these soleus muscles that were exhibiting a chronic activity pattern or a tonic activity pattern now are not being used as much and are actually in effect unloaded. And if you don't use it, you lose it. So that principle applies to spaceflight, but also applies to ground-based models of unloading as well. So if you don't use it, you lose it. We know that you lose muscle mass, you lose muscle strength. This is a cartoon depiction of a healthy muscle section. If you go up in space for a period of time, you'll see that the fibers become smaller, they become angular. It's almost depicting sarcopenia. In fact, some would consider that spaceflight is a rapid model of aging. But what I want to talk about today is that if you don't use it, you lose it. That principle not only applies to a reduction in muscle mass or a reduction in muscle strength, but it also applies to a reduction in the Circa function or in its capacity to bring calcium back into the SR. So over on the right here, we have two models. These are from murine models, so two mouse models. So the first one's denervation. So this is a model of disuse. And this is some work that we did when I was at the University of Waterloo in collaboration with a group in Ottawa, Jerome Furnett. And what we're showing here is that maximal Circa activity on the y-axis is significantly reduced in the soleus muscle after denervation. Over on the right is the Tanotomy model. So this is a model that we use quite frequently in my lab. Essentially, we unload the soleus muscle by transecting the soleus tendon. And when we do this 2 weeks after, we see, sure enough, there's a significant reduction in Circa activity, its ability to hydrolyze ATP. In humans with unloading, there's also evidence for altered calcium handling. And this is a study that was published earlier this year, led by Dr. Elena Monti from the University of Padova, and my colleague here, Carlo Reggiani, was the second author here. And what they did was really interesting. So they used bed rest, and bed rest is a Earth-based analog of spaceflight, imperfect, of course, as with any spaceflight analog here on Earth, but bed rest will still unload the muscles. And what they found is that with 10 days of bed rest, there was altered intracellular calcium handling. So to do this, they used the skin single fiber technique. And this slide's a bit busy, so I apologize for that, but I'm going to try to go a little bit slow here. And just to give you an idea of skin fibers, this is actually a fiber that I skinned a while back. And this is a technique that I learned at La Trobe University in Melbourne, Australia with Dr. Graham Lamb. I'll just pause it here. And so this is a single rodent fiber, and oftentimes these fibers are 50 to 100 micrometers in diameter, less than a centimeter in length. So this is all done under the microscope with forceps. In this case, it's called mechanical skinning. And so what I'm doing here, I'd like you to imagine as if you're wearing a long sleeve and you roll that long sleeve up. When you roll that long sleeve up, you're exposing your forearm. And essentially what we're doing here, we're exposing the fiber. We're gaining access to that myoplasm. So this cuff right here is the sarcolemmal membrane, the plasma membrane. We're gaining access into the myoplasm. Now, what Dr. Elena Monti did was a little bit different. They used chemical skinning, so it wasn't with forceps. They used chemicals and detergents to get rid of that sarcolemmal membrane, but still, nonetheless, they're trying to gain access into that myoplasm. And what they do is they'll take that skin fiber and they'll mount it. This is actually the setup from Dr. Graham Lamb's lab in Melbourne. And you can see that you mount that skin fiber onto mounted forceps, but also onto a force transducer. And when you do this, you can expose that skin fiber because you've gotten rid of that membrane, the covering. You can expose it to different solutions to indirectly measure SR calcium handling. And essentially what they're trying to do is they're going to take that skin fiber, this little fiber portion here, and they're going to load it with calcium. So they're going to place it in a bath called loading solution, which has calcium. It also has ATP, right? Because the Circa pumps are going to use that ATP as energy to bring calcium into the SR. And they're going to load it for 5 minutes. And then after that, they're going to place it into another bath using this laboratory jack, for example. And this other bath is what they call caffeine release solution. So right next to it, let's imagine it's a caffeine release solution, and it has 20 millimolars of caffeine. That's quite high. And that caffeine is going to open up these orange ryanodine receptors. It's going to open them up, allowing for most of that calcium to get out of the SR to elicit force production. And because that fiber is mounted onto a force transducer, you're able to measure the amount of force produced. So it's an indirect measure of SR calcium handling. If the muscle fiber is better at loading calcium into the SR, then more will be released and more force will come of that. And of course, they normalize their force to max force production. I won't go through that. But what they found was that after 10 days of bed rest, so I'll orient you to this graph, they're looking at type 1 fibers and type 2 fibers after 10 days of bed rest in humans. And this, again, this is actually in the supine bed rest condition, in case any of you were wondering. So baseline or bed rest 0 is shown here in the open bars, and 10 days post bed rest is in gray. And if you just focus your attention maybe on the type 1. So they did this in 2 trials. They loaded the SR, released the SR, loaded the SR, and then released the SR. And then they also had a total. I won't talk about that in too much detail, but nevertheless, what you can see is that the ability of these fibers after bed rest to take calcium into that storage site and then release calcium out of the storage site and elicit force was significantly reduced across both type 1 and type 2 fibers. So a really, really interesting technique that they use there to show that there is SR calcium dysfunction with muscle unloading. And so what is the underlying mechanism? This is a highlighted bit that I took from their discussion, and I'll read it here. The most likely mechanisms responsible for the functional impairment of the SR are an increased leakage of calcium out of the SR. So that just means it could leak out of the RYR, and an impaired capability to pump calcium back in the SR. And again, I have this actually underlined here because again, the ability to bring calcium into the SR depends on these Circa pumps, okay? And actually this will help me transition back to Jessica's study. And the purpose of this study, you know, with all of this in mind was, okay, if there is impaired calcium regulation at the level of the SR with unloading, well, let's see what happens with spaceflight. And unfortunately, we didn't have any muscle biopsies from human astronauts. I really wish we did. I still wish I could get my hands on some, to be completely honest. But nevertheless, we were able to get mouse soleus samples from male and female mice from NASA's biospecimen sharing program. And what Jessica sought out to do, again, in this side project of hers, was to examine what happens to Circa function after a period of time in space or exposure to microgravity. And before I talk about her data, I will say that mice that are up in space or exposed to microgravity will experience similar things in comparison to humans. We'll see muscle atrophy and a slow to fast fiber type shift, particularly in the soleus muscle. So look, this is looking at 91 days aboard the International Space Station. F stands for flight, G stands for ground control, L stands for lab control. Actually, this video should play, so just give me a second here. So when the mice are up in space, I'll pause it here, they're in these specialized cages, and you can see it's wired all the way around so that they have something to grab onto. Their food bars are there. So L is lab control. So essentially it's the mice are housed on Earth in their standard lab conditions. The ground control, the mice are housed here on Earth, but in this specialized cage. The flight, or F, is the mice are housed in these specialized cages, but are of course up in space. And you can see that fiber cross-sectional area is reduced in the soleus, as we'd expect. If you don't use it, you'll lose it. But there's also a slow to fast fiber type shift. You see that the soleus muscles, which are predominantly comprised of these oxidative fibers, which rely on oxidative phosphorylation, they're generally fatigue resistant. And in a sense, you would kind of want that, right? As a postural muscle that's on for a longer period of time to withstand that never ending downward pull of gravity, you're gonna want fibers that are generally fatigue resistant. So then if you don't use it, you'll lose it. What actually ends up happening is these fibers will transition from a more oxidative and fatigue resistant fiber type to now a more glycolytic and less fatigue resistant fiber type. So we see similar things with humans. I don't have the human data here, but we do see this similar thing. So it just means that we can use the mouse model as a model to examine the cellular mechanisms underpinning these changes in muscle, such as Circa function. So what happens to Circa function with spaceflight? So the first piece of data I'll show you is from male mice. In this case, they were exposed to microgravity for 37 days and part of the Rodent Research 9 mission. And so similar to what I just showed you in the last slide, there's a couple of different controls. We have the ground control, which are housed in those NASA cages, but here on Earth, and then the vivarium control, which are just housed in standard lab cages. And what Jessica's doing here is what we call a calcium uptake assay. So we're trying to measure the ability of the Circa pumps to bring calcium into that storage site, the Sarcoplasmic Reticulum. And to do this, we use a calcium fluorescent indicator. It's called INDO1. Now, the thing about INDO1 is that it can't pass the SR membrane. It can't go through here. So in effect, what it does is it measures cytosolic calcium levels. So when we provide the muscle samples with ATP, that energy, and that energy is going to be used by Circa to bring calcium into the SR, then cytosolic calcium levels should drop. And you see that quite nicely in the ground control and the vivarium control. But you don't see that here over time in the flight group, suggesting that Circa's ability to bring calcium into the SR is impaired. There's a lot more calcium staying out in the cytosol. And we see the same thing happening with female mice. In this case, they were exposed to microgravity for 35 days. And part of the Rodent Research 1 mission, we also have the ground control and the vivarium control. Again, when we add that ATP, which Circa is going to use to bring calcium into the SR, we see that drop in cytosol calcium in the two controls on Earth. But in the flight group, we don't see that. So both in male and female mice, we see impairments in Circa function. And so Jessica then asked me, well, why is Circa function impaired? And she ran a battery of Western blots. So essentially these Western blots were looking at protein content. And the first thing that she looked at was Circa content. And what's interesting is that we found that there was a significant increase in Circa 2A and Circa 1A. These are just two isoforms of Circa that are dominantly found in muscle, skeletal muscle. So the flight group here is in red. You can see an upregulation of Circa 2A and Circa 1A. And we think that this was a failed compensatory response. So our muscles are trying to increase Circa protein, Circa enzyme, to compensate for the fact that Circa function is impaired. However, this was failed, as you could see, as you saw in the last two slides. And the second thing that Jessica looked at were the expression of these circumregulatory proteins. So phospholambin, sarcolipin, and a new one that Jessica actually discovered in skeletal muscle called neuronatin. And this is actually part of her PhD work. And if you want to learn a little bit more about how neuronatin regulates the Circa pump, here's her publication in Feb's Letters that was just accepted a few weeks ago. So she's on fire. But anyway, these regulatory proteins, phospholambin, sarcolipin, and neuronatin, they all will bind to the Circa pumps. Okay, they all physically interact with the Circa pump. And when they do, they inhibit its function. So when she took a look at their expression or their protein levels, we found that phospholambin content was reduced, sarcolipin was upregulated. Now, this wasn't really surprising given what we know about sarcolipin in terms of previously published literature. I'm not going to get into it today. And then neuronatin was actually reduced. So in terms of Circa content and circumregulatory protein, the protein contents of these phospholambin, sarcolipin, and neuronatin proteins, nothing really stuck out to us in terms of the culprit causing these impairments in Circa function. So finally, Jessica looked at markers of reactive oxygen nitrogen species. So these reactive oxygen nitrogen species, they're going to steal electrons from lipids, from proteins, these enzymes that will end up damaging them and impairing their ability to perform. So what Jessica ran is what we call nitrotyrosine and nitrocysteine. So total protein nitrocysteine and total protein nitrotyrosine. So these are just markers of oxidative and nitrosative stress. And I've got the blots here to show you quite clearly that in the flight group, shown here in F, there is a significant upregulation of total protein nitrocysteine and total protein nitrotyrosine, suggesting that there is this elevation in oxidative and nitrosative stress. Now, this is important in the context of Circa because the Circa pumps themselves are very susceptible to damage caused by oxidative and nitrosative stress. So this is, again, Jessica's published work. This was actually her published work as an undergraduate student in my lab back in 2019. And what she did was she looked at the cardiac muscles from a model of Barth's syndrome. So Barth's syndrome is this X-linked recessive disorder characterized by cardiomyopathy, but more importantly, mitochondrial dysfunction. And with that mitochondrial dysfunction is an increase in reactive oxygen and nitrogen species, what we call RONS. And she looked at the ability of Circa to perform in these hearts. She looked at them at 3 to 5 months and 10 to 12 months. And so in this case, it's not a calcium uptake protocol, but this is a Circa activity protocol. So on the x-axis, we have calcium concentration shown in PCA units. So it's a little bit backwards, it's like pH, it's the negative logarithm of the calcium concentration. So just know that as you go from left to right, you're increasing calcium concentration. And in the wild type hearts, so otherwise healthy hearts, you're going to see that as you increase calcium, Circa is going to increase its activity because it's seeing this calcium it's being exposed to this calcium, well, it wants to bring it into the SR, so it's going to increase its activity. You see that quite nicely in the wild type. You don't see that as nice in the TAS knockdown. And this happened both in 3 to 5 months and 10 to 12 months of age. And the reason behind this was, again, because of this increased production of reactive oxygen and nitrogen species. In fact, when we look at Circa protein specifically, and we look at protein modifications, in this case, Circa 3-nitrotyrosine, which you only see elevations of this if there is oxidative or nitrosative stress. And you can see significant elevations in 3 to 5 months and 10 to 12 months of age in the heart. So this was actually co-immunoprecipitation. We weren't able to, or Jessica wasn't able to run this co-immunoprecipitation with the Spaceflight sample simply because of tissue limitations. But nonetheless, we do think that the impairments in Circa function are likely due to elevations in oxidative stress. And I really like this schematic that Jessica has devised because it shows this vicious cycle. So mitochondrial dysfunction can increase the production of reactive oxygen and nitrogen species, which, by the way, can contribute to muscle weakness and atrophy through pathways that I'm not going to get to touch on today. But as I just showed you, these reactive oxygen and nitrogen species will damage the Circa pumps. Now, the kicker is when you damage the Circa pumps, you're going to impair their ability. You're going to impair their abilities to bring calcium into the SR, that calcium storage site. So in effect, you're going to raise intracellular calcium levels. And chronic exposure to elevated intracellular calcium levels will contribute to mitochondrial dysfunction, will accelerate reactive oxygen species and nitrogen species production, will accelerate Circa impairment. So you get this vicious cycle. And what we're wondering here is, maybe we can stop the cycle by improving Circa function. If you improve Circa function, can you mitigate the mitochondrial induced production of reactive oxygen species and therefore the ensuing muscle atrophy and weakness? And if we go back to this schematic where spaceflight causes muscle unloading and disuse, and even here on Earth in other conditions of muscle unloading and disuse, they all operate under this premise of if you don't use it, you'll lose it. You're going to lose muscle mass. You're going to lose muscle strength. And as I showed you today, you're going to lose muscle Circa function. So the question is, if you improve this, if you improve Circa function, can you attenuate the reduction in muscle mass? Can you attenuate the reduction in muscle strength? And maybe it's something that can be combined with exercise prescription. Maybe it's something that we can add with exercise with the astronauts up there in space to further try to dampen the effects of unloading on muscle mass and strength. And there's a little bit of precedence for this. This is the last data slide that I'll show. And it's not from our group, it's from a different group published last year showing that pharmacological activation of Circa in aged mice, so 24 to 26 months of age, will attenuate the muscle atrophy and weakness that you see with aging. So here in this first panel over on the left, they're looking at 16-month-old, so young, versus untreated 24 to 26-month-old mice. And you can see a significant reduction in Circa activity. However, when they treated these mice with an allosteric activator of Circa, it's called CDN1163, and they treated them for 8 to 10 months. You see that effect of reducing Circa activity was prevented, as you'd expect. But over in the middle panel here, you see that when you look at 16 months old versus 24 to 26-month-old mice untreated, you see a significant reduction in gastrocnemius muscle mass relative to body mass. So that atrophy. However, that atrophy is prevented when you activate Circa function. And in terms of force production, if you're looking at the panel B here over in the on the far right, this is actually looking at the extensor digitorum longus. So you see that specific force is declined in age mice 16 months versus 24 to 26-month-old. However, this decrement is prevented with an improvement in Circa function. And the point here is that I made it before, is that spaceflight may be considered a rapid aging model. So whatever we learn in spaceflight in terms of cellular mechanisms may be applied to aging here on Earth. But perhaps the reverse can be true, and whatever we learn from aging can be applied to space flight. For example, pharmacological activation of Circa or improving Circa. So future studies from our lab will be looking to improve Circa function to see if that mitigates the muscle unloading induced atrophy and weakness with hind limb suspension. So we'll be looking at pharmacological activation, but also genetic manipulation, things like overexpressing Circa. And so if there's anyone in the audience there that's interested in joining our lab, please feel free to contact me if you want to tackle this project. Because if I don't get anyone to tackle this project, I'm afraid that Jessica might try to do it, and she has her own PhD work to do. Anyway, I'll leave it there, and I'll say thank you to my lab. Thank you to Dr. Chung and the committee for inviting me here to give this presentation. Thank you to our funding sources, and of course, NASA for providing us with those samples. And I'll be happy to take any questions.
Well, great. Thank you so much, Val. And you forgot to plug our heat therapy project that we're just about to start.
That's right. That's right. With muscle unloading with that as well. So much going on. I'll have to come back and give it. Oh no, you'll have to come back and give this talk.
Maybe, but I need help learning the lingo a bit more first. But I'll start off with some very naive questions, since again I don't work in animal models. So maybe you can help me and some of the audience members out probably too. But also some of these are kind of related to kind of naive questions about the impact of space flight and return on Earth. Most of your discussion so far has been on atrophy during space flight. Have these kind of equivalent studies been done in terms of what happens when after space flight, and trying to, again, I guess you touched on it at the end in terms of how quickly do the kind of the atrophy or the loss of Circa function, et cetera, kind of come back, if they do at all, post space flight or post bed rest? Any insight on that?
That's a really good question, because you know, when you go up in space, at some point there's going to be a return to gravity, whether that be full gravity or partial. We're talking about further space exploration. But unfortunately, I don't think there's any data there looking at Circa function specifically. But it is a really good question, because when you return to gravity, there is going to be an acceleration, in my opinion, of reactive oxygen and nitrogen species, at least for a small period of time. It's almost like ischemia reperfusion. So, but I think maybe, this is just a guess, if you were to improve Circa function chronically, you would be better at this functional return, what we call functional return. So unfortunately, there's no data there, but that is a logical next step.
Okay, great. And we have a question in the Q&A that was almost going to be one of my follow-up questions. The other thing is, when you talk about Circa function decreasing, I mean, is it a loss of kind of capacity or the muscle's ability to use them, or is it kind of a wholesale, the Circa channels just disappear and kind of gets reabsorbed? Is it kind of loss of the units themselves or loss of ability to use them?
That's also a good question. So with Jessica's study, we did see an upregulation of both Circa isoforms. Now that was in male mice. In female mice, we actually only saw an upregulation of one Circa isoform. So there is a potential there for a difference in terms of biological sex. But the increase in Circa, we believe, is not functional. I think the reduction in Circa function is due to damage caused by these reactive oxygen and nitrogen species. And perhaps the elevation in Circa that we saw with the male mice is due to an inability to clear damaged proteins. So our muscles will have this innate program to clear dysfunctional proteins and enzymes, but maybe that is off with spaceflight. And so I do think that the impairments in Circa function are caused by damage to the pumps rather than any change in their content.
Okay. And Berekit has a question of Berekit Falk. There seems to be a similar effect of spaceflight on type 1 and type 2 muscle fibers. I guess the question is really, do the changes in Circa function, are they similar in type 1 and type 2, or is it accelerated in type 2 because they tend to be more kind of fast and kind of obviously rely much more on a really fast sarcoplasmic reticulum?
So yeah, that's a good question. So I would think that, I mean, without having this single fiber data, being able to assess Circa function and the ability to take calcium in with Spaceflight samples, my guess would be that the Circa function is being impaired across both fiber types, but because there is a slow to fast fiber type shift, then just by proportion, you're seeing more impairments in the fast fibers. But that is interesting, right? Because on Earth, in other conditions, in other healthy conditions, if you undergo a slow to fast fiber type shift, you will see an acceleration of Circa function, or you'll see an enhancement of Circa function. So one thing I didn't show was that we also had samples, muscle samples from the tibialis anterior, and they're not as affected with Spaceflight compared to the soleus. So when we looked at the TA samples, we saw that slow to fast fiber type shift, but we didn't see any changes in reactive oxygen and nitrogen species. There was no elevation in this oxidative or nitrosative stress. So in opposite of what we found in the soleus, the TA samples that were up in space, this was published in Jessica's paper, the TA samples after being in space showed an acceleration of Circa function, an improvement in Circa function, because it was just a slow to fast fiber type shift in the absence of an increase in reactive oxygen and nitrogen species.
Okay, and maybe the final one from Rafi Dotan is, yes, what you've talked about so far is really kind of bed rest or immobilization type, and how much similarity is that kind of a scenario with detraining? And if you had a very fit individual and they just kind of completely stop exercising but are still obviously like active and in terms of being mobile and living, is that kind of just similar or different in shades of gray, or is it kind of, or is bed rest and this immobilization in space kind of completely night and day different from a detraining model?
I think, in my opinion, this model with bed rest and spaceflight might be a little bit more extreme. So the effect on Circa function might not be as apparent. It might be there, but maybe not as apparent. I don't have the data to back up that claim. But I also think that detraining in terms of Circa function might have more of an effect, however, in cardiac Circa function. So with training, we know that cardiac Circa function will improve because there's an improvement in or increase in Circa content. And I do believe that reverts with detraining. So with the skeletal muscle and in comparison to bed rest, I do think that they are, there's one model that's extreme and then one model that's not as extreme, if that makes sense.
Okay, and then this will be the last question from Shonda Morrison. And are there countermeasures you could do pre-flight to minimize the problems observed with the Circa function in space? So, for example, nutritional, or again, we just mentioned heat therapy, or kind of loading up on exercise pre-flight, kind of in the same idea of now individuals pre-having before surgery or before kind of injury. Is there anything that can be done, or kind of can you super stop kind of Circa function?
I think you could. I think, what you're talking about with heat therapy and even exercise prior to, you're going to increase the expression of something called heat shock protein 70. So it's a chaperone protein with heat therapy, also with exercise, regular exercise. Dr. Peter Titus has shown this with his program, that regular exercise is going to increase heat shock protein 70. HSP 70 will bind to the Circa pumps and will protect them in conditions of elevated oxidative and nitrosative stress. So that is a really good question, whether there's this prehab, and maybe it is heat therapy, maybe it's regular exercise, maybe it's both. to preserve Circa function or maintain Circa function before they go up in space? Really good question.
All right. Well, great. Thank you so much, Val. That was very, very interesting talk, and I can see the fascination with mice models, although I'll still stay mostly on this side of the human model work instead, though, for now.
Well, thank you.
All right. So if you can stop sharing your screen. But yeah, and congratulations on the lab and all your students. They're just
Thank
all
you.
awesome.
Yeah,
All right.
they really are. It's easy for me.
Yeah, so we will switch now to Dr. Eric Rivas from Johnson Space Center. And again, thankfully, he has managed to get clearance to speak, and he's going to be talking to us about kind of more macro human big picture in terms of what actually happens in space, in terms of exercise countermeasures, and also some of the work that goes on in Johnson Space Center and life sciences. So Eric, if you wish to share your screen.
Yeah, give me a second. I'm trying to figure this. Are you able to see my screen?
Yes, right now we're seeing kind of the whole PowerPoint.
Okay, yeah, let me zoom it out. All right.
There we go, perfect.
Okay, so the title of my talk is Exercise Countermeasures for Spaceflight. I'd like to first thank the organizers and the committee for the invitation to talk more about what we're doing at NASA. Briefly, I just go through the outline as I walk you through this presentation. I will cover a little bit about the NASA Human Research Program, go over some of the risk of spaceflight, talk about the historical progression of the exercise countermeasures from the start till where we are now, briefly go over the exercise requirements and show how over time we've changed the exercise hardware and how that affects the exercise prescription. And then lastly, the exploration exercise devices and how that will impact how we prescribe exercise and maintain crew fitness and health. So the NASA Human Research Program, it is comprised of 5 elements, and these are the 5 hazards that NASA is particularly concerned about for the astronaut in space: space radiation, isolation, the distance from Earth, gravitational fields, and hostile closed environment. Primarily, the major research involves the International Space Station medical projects, space radiation, human health countermeasures, medical capabilities, and human factor and behavior performance. So from that broad theme, I'll narrow it down to the human health countermeasures. And this was talked briefly on one of the questions. So we do look at the different phases of spaceflight. For this, the particular overview objective is to really develop and assess medical standards, vehicle spacesuits requirements, and countermeasures. And this entails pre, in, and post-flight countermeasures. And here we list a couple of examples. So for pre-flight, we look at the candidate profiles. We have medical standards for fitness that we assess, and then we measure them during flight and post-flight so we can carefully evaluate the change in fitness over the duration of spaceflight. The 5 portfolios we have are vision and cardiovascular, exercise and performance, multi-system, bone, and technology and infrastructure. So from that program, I will narrow it down to the lab that I work in is exercise physiology and countermeasures. As you can see here, we're not all physiologists. We are a team of biomechanists and engineers and data managers. And we look at all areas of spaceflight for improving astronaut health and fitness. Particularly, we look at the risk and gaps, and we design research to answer questions that minimize the risk of the crew. We assess countermeasure system and operations. I mentioned this previously. We have developed medical testing for fitness that we do pre, during, and post spaceflight. We also evaluate hardware, doing human-in-the-loop testing, and we develop task standards development, and we also look at new technology as well. Okay, the risks for spaceflight. Some of the risk was mentioned in the previous talk for the muscle deconditioning, and what was exactly what was mentioned previously. So when we move from 1G to 0G, when we remove gravity on Earth to space, this causes the deconditioning of major organs. This deconditioning really affects the functional impact that the crew need to be able to perform in space or when they come back to Earth. So it affects the ability to perform critical tasks such as emergency egress. So when the astronaut comes back to Earth, they need to be capable of removing themselves from the capsule. They need to also perform critical tasks such as extravehicular activity, or if they're reintroduced to a gravitational environment such as the lunar surface, which is 1/6G, they have to be able to perform there. And if these major organ systems are deconditioning, that increases risk of injury or even life. And then lastly, long-term crew health is a major concern. So the risk of spaceflight on the skeletal system, I will walk you through this, this figure here. So the y-axis shows the change from pre-flight, and this is bone mineral density. The x-axis is representing the different bone regions of the body: the lumbar spine, femoral neck, trochanter, total hip, and the pelvis. The gray circles is the MIRS mission, and the triangle R is the ISS, and the line is just the means. And as you can see here, there's large variability in the different locations of the bone, ranging from 21% gains to nearly 5% losses, depending on the specific skeletal site. And the major cause of this is obviously the unloading on the bone, and this causes an excretion of calcium in the urine and a decline in bone mineral mass and quality. And this is after 6 months of spaceflight. And the reduction in bone mass and quality will increase the risk of bone fracture. So the risk of spaceflight for skeletal muscles, as was mentioned previously, we have atrophy due to of the muscle volume, and this causes a reduction in type 1 and type 2 fibers. This data here shows on the top left 17 days of spaceflight in the gastrocnemius and the soleus, nearly a 10%, but there's more so the longer you go into the mission. So 16 to 28 weeks, you see nearly a 20% drop in muscle volume, and you can see the fiber size changes on the lower right, pre to post flight, and the fiber distribution of type 1, type 2A, and type 2, which is the white, gray, and black filled in bars. And then this, obviously, the reduction in type 1 and type 2 fibers reduces maximal voluntary contraction and reduces muscular endurance. This is data from the Skylab mission, and I apologize for the blurred image. This is from 1977, so we have data from way back on the Skylab mission. This is a 27-day mission, and I'll just walk you through it because it's a little bit difficult to understand. So the black box is the weight, the diagonal box is leg strength, and the open is leg volume. The top part is the cycle ergometer intensity, so watt minute per day. So they exercise at about 30 watts minute per day on the cycle ergometer. And the lower portion of this figure shows the weight and volume and percent loss. And you can see here leg strength loses about 25% reduction. And this is really a sort of a control experiment here, because this mission here was a 28 mission. It did not have resistance exercise capabilities. And you can see that the drastic loss in only less than 30-day mission. This data here shows the time course and percent change in leg strength. And this is for the knee extension for bed rest studies. The y-axis shows the percent change in strength, and the x-axis shows the days of bed rest. And this is accumulation of many bed rest studies. And you can see the linear drop in strength for the bed rest studies. I've highlighted the most recent one that we published, the Pult-Snyder exercise, which also had a control no exercise. And you can see the red dot was within that narrow range in reduction for a 70-day bed rest study. However, the exercise was semi-protective, as it didn't fully protect it, but it maintained some strength over the 70 days of bed rest. So the major risk here is reduced capacity to perform long duration extravehicular activity and capsule egress if an astronaut crew reduces muscle strength. Now, spaceflight risk on the cardiovascular system. You can see here in this figure, it shows you the difference in pressure from the head all the way down to the feet. And this is on Earth, 1G. However, when you move from 1G to 0G, you see the pressure normalizes across the body. This causes a shift in blood volume to the upper body. And when they come back post-flight, it causes a reduction in blood volume, red blood cell mass, and reduced plasma volume. And this causes orthostatic intolerance. And most likely this is due to the baroreceptors sensing that increase in pressure change, which causes the bodies to get rid of fluid. And we have this little figure here that shows the baroreceptors addressing the change in pressure. This data here supports the increased risk of orthostatic intolerance, moving from supine to upright change in position. And this is after 16 days of spaceflight. You can see there's a decrease in stroke volume, which here on the figure here, y-axis is muscle sympathetic nerve activity in bursts per minute, and the x-axis is stroke volume in milliliters. This change in position from supine to upright shows a decrease in stroke volume and increase in sympathetic nerve activity. And during the supine to upright positions after 16 days of spaceflight, it also significantly decreased stroke volume and increased sympathetic nerve activity. The major risk here, again, as I've mentioned, the reduction in blood volume, the changes in this adjustment to blood pressure and the pumping of the blood of the heart reduces the risk of orthostatic intolerance and reduces the capacity to perform long duration extravehicular activity and capsule egress. And then lastly, the risk of spaceflight on the cardiovascular system. So we measure aerobic capacity through a psychoergometer peak test. Here on this figure here, on the y-axis shows the VO2 peak in milliliters per kilogram per minute. And on the x-axis shows over the duration of the spaceflight. L-3, 1 month is 3 to 1 month before liftoff. FD is flight day 15. FD is flight day 75. R-14 is 14 days before return. And then R+3 is when they are on the ground, 3 days on the ground, and then 30 days post space flight. And you can see over the duration of from pre to post, there's about a 10% reduction. And this is even with the current exercise countermeasures. And you can also notice that there is high variability of those that really improve and those that really lose fitness over time. And these dotted lines are just representation of these the proposed standards that we're developing currently, where we assume that if there's greater than 20% fitness, then there's a high probability of task failure and increased risk of crew health. And then greater than 30% is high probability of the loss of the objectives of the space flight. And most importantly, the risk is reduced capacity to perform long duration extravehicular activity and capsule egress. Now, briefly, I'll go over through the historical progression. Let me just walk you through this figure here. The blue circles show the mission. The lined text is the exercise hardware, and the bulleted italicized text is the exercise prescription. So you can see here early Mercury and Gemini missions, there were no hardware available, there was no exercise prescription, and they just used a bungee cord for 30 seconds. And really during this time, this was really the first time we did exercise in space, and they wanted to look at the heart rate and blood pressure response. In the Apollo mission, we had an exercise genie and a cycloergometer, but no standard program. They exercised for 15 to 30 minutes per day. And then the Skylab's mission is when we increased the capabilities, included a cycloergometer, a treadmill, MK1, MK2, and I'll go over that more in the next slide. But during this Skylab 2, the crew really wanted an increase in exercise in the exercise program, both in time, duration and time. And they increased exercise time duration in the Skylab 3 for 1 hour a day. And then Skylab 4 mission, it was about 1.5 hours a day. And we added the treadmill during that time. In the shuttle and the shuttle mirrors program, we had the cycloergometer, we had a passive treadmill, an additional EDO treadmill, and a rower. And you can see here as we progress over time, every mission, we're improving the exercise capabilities, increasing frequency 3 days a week. And the intensity was 70% of the age predicted maximum, and about exercising 20 minutes per day. And then ISS to current present, we have the CVIS, TVIS, IRED, ARED, and T2. And these devices are described down here: advanced resistive exercise device, cycloergometer with a vibration oscillation stabilization. IRED is the first resistance exercise device, the interim resistance exercise device, and then the treadmill, the 2 treadmills, one in the second version. But most importantly, you can see here now we have an organized approach to train our crew. We have trainers called ACERs, astronaut strength and conditioning rehab specialists, that train our crew and monitor them throughout the duration of spaceflight. And we have 6 to 7 days of frequency for resistance and aerobic exercise now. Prescribing exercise, it's not only about the variables of exercise prescription. We have to really think about the hardware and how the hardware and the vehicle integration impacts the prescription of exercise. So for frequency, intensity, time, volume, progression, the capabilities of the hardware determine how well we can prescribe exercise. So the hardware needs to be compact. It needs to provide a robust stimulus for aerobic and resistance exercise. And it also has to provide variability of exercise movements. It can't just be one movement over and over. The crew would get bored by doing that. And then also the device has to have a vibration isolation system. It has to fit in the operational volume. And it also has to have an environmental control and life support system. And this system buffers for CO2 levels, humidity, and temperature of the exerciser. And then, as you can be aware, if the ECOLIS system isn't that, the capabilities of that is lacking, then that will affect the exercise that the crew are doing. That we would have to have them have more time in between each exercise session so the ECOLIS system can catch up. And ultimately, these 3 major variables would protect against the major organ systems and protect against fitness for extravehicular activity and egress. Now, how these have changed over time, I can walk you through this. These are the Skylab missions 2, Skylab mission 3, Skylab mission 4. You can see the Skylab mission 2 was 28 days, only had a cycloergometer. The exercise intensity was 31 watts per minute. And you can see the change from pre to post flight was 0.8% reduction in muscle strength per day. When we added the cycle, when the cycle was added, the MK1 and MK2, it decreased from 0.89 to 0.44% per day. And you can see here to the right, the MK1 and MK2 were really just springs that were, that were a pulley system that had about a maximum force of 25 pounds per foot. And then when we added cycle, MK1, MK2, and treadmill, you can see the reduction in the strength was 0.09% per day. And again, these are different flight durations, but when we normalize it to 30-day mission, you can see the progressive improvement in exercise capabilities to protect against muscle strength. The first resistance exercise device was the Interim resistance exercise device, and this is on the far left. The concentric load was up to 300 pounds. The ratio of eccentric and concentric was 60 to 80%, but the load was not constant throughout the range. And then we have what we have now is the ARED. It increased the concentric load up to 600 pounds. The eccentric concentric ratio was about 90%, and it was a constant force throughout the range of motion. And you can see here for bone density, the ARED versus the IRED, which is the gray, compared to the Russian mirrors, which had no resistance exercise capabilities, which is at the dark gray. You can see the rapid improvements of using the ARED compared to the Russian mirrors in the IRED. And the same thing here for the isokinetic strength from pre to post space flight for the knee, ankle, and trunk, comparing the ARED and the IRED. Improvements of protecting against strength and bone measures. Briefly, these are the exercise requirements for the ISS mission duration, or the long duration mission. Frequency is 6 to 7 days per week. Total exercise time is about 60 minutes on the ARED, and for aerobic exercise, it's about 30 minutes a day, and we use high intensity interval exercise on the Treadmill 2 and the Rivas cycle ergometer. So this is in the works, and we're looking at improving the exercise prescription. So I'm presenting here really just means here, but we're in the process of downloading, reviewing all the data that ISS has and running some statistical modeling to show which are the best predictors for exercise performance. But these two studies have been published, the English and Moore paper. The English sprint study looked at high intensity exercise and reducing frequency of resistance exercise and has shown promise. But let me work you through this table here for exercise prescription. I just titled them A, B, and C. A is the Moore paper data, and B and C is the English, and B was the standard of exercise. C was the sprint, increasing intensity, reducing the frequency for resistance exercise. But the difference between the two here was that A and B, the comparison between A and B, A exercise at about 74% of their peak heart rate, whereas B and C, they exercised at a higher intensity, and then B and C included high intensity interval exercise. And you can see in the figure up here, the change from pre to post flight. The black is the A, and the white and the gray is the B and C. And you can see the rapid change from about 15% to about 5 to 10% in maintaining fitness, or the percent change from pre to post space flight. And this was 6 months duration mission. Similar findings for aerobic capacity for looking at peak workload and then ventilatory threshold. There was no difference between the two, suggesting that we can reduce volume, but we need to increase intensity of exercise to maintain aerobic capacity measures. The same thing for resistance measures. The sprint study showed that when we compare to the standard of exercise prescription, if we increase intensity about 6 to 15% and reduce the time spent doing resistance exercise from 6 to 7 days to 3 days per week, we're able to maintain the same level of strength from pre to post 6 month space flight. And then lastly, most importantly, the exploration exercise devices. This figure here shows where we're at now, what we're using here at the International Space Station. But NASA has big goals to go back to the moon and then moreover go to Mars. But that's going to require moving from the ISS to the Orion and Gateway. And as you can notice, the volume within those vehicles is extremely smaller, and therefore the exercise capabilities will be reduced. So there's challenges within the small vehicle. It's highly restricted in volume, mass, and power. The next generation of hardware exercise devices has to combine aerobic and resistance exercise, which will cause possible performance limitations. And you can see here in the lower right cartoon, the device that's being developed that'll fit in this capsule. And then here's, again, more of what we're designing here. We have the limitations for the exploration exercise devices. They have to provide the capabilities as what we are using on ISS, but we may be limited in that. So we have to think of novel ways to maintain fitness with exercise prescription, and at the same time figuring out ways these small compact exercise hardware can provide the robust stimulus for aerobic and resistance capabilities. And that is the end of my talk. Thank you, and I can take any questions.
All right, thank you very much, Eric. So again, please type your messages into the Q&A and I will moderate them. So Eric, can you just talk me through, I guess, you do have the plan to go to the moon and to Mars, and again, is it just going to be trying to get things like ARED, whether it's smaller, kind of lighter, more compact, or where are the thoughts right now in terms of the exercise kind of equipment? Is it going to be, again, just miniaturization or something else?
Yeah, it won't be ARED. It'll be something like this device on the lower right, which is, I don't know if you're familiar with the flywheel device. It's isoinertia. It doesn't require energy or power, and it provides a robust capability. And yeah, something like this is what we're planning for.
Okay. And yeah, certainly the data you've shown on kind of sprint type or high intensity training seems promising too, if that can reduce the kind of the time demands for exercise training. Have that really been, how far along is that in terms of being further validated as a kind of being regularly prescribed in current ISS missions?
Yeah, they're starting to move in that direction. But like I said, I think our next step is, we did one study with the sprint study that did a bed rest, and then they did the ISS 6-month mission, which is the data I presented there. But I think we still need to go back and analyze the data that we have to kind of really see what's going to work. But the other problem is that all that data and those studies were on ISS capability hardware. When we move to exploration devices, we don't know if it's going to provide the same protection. So we need to come up with novel ways to maintain fitness, crew, and health. And that may include pharmacological approaches as well, combining that with the exercise.
Right. Thank you. And Bernadette Falk has a question. In one of the slides, you demonstrated that the addition of treadmill exercises on one of the earlier missions prevented strength loss. So the first question was which muscle groups were tested? And then the second part of the question was, it makes sense that the addition of resistance exercise in later missions prevent strength loss, but how does treadmill exercise prevent strength loss, especially if there's not really that much kind of gravity loading?
Yeah, that was really surprising, but I think it's the addition of the treadmill, the straps, like you can see here on the right side, the strap loading on the body produces a load on the bone and the muscle. And the second question too, what muscle groups? We typically use the leg when we measure changes in strength. Typically, we don't see loss in volume of muscle in the upper body. We typically see it in the lower body. So we're more concerned about the lower body reductions in strength and volume, muscle volume.
Right. Chris,
and this may be due to the fluid shift into the upper body. There's probably less nutrients that are getting sent to the lower limbs.
Kristen, today, any follow-up questions?
Hi, Eric. I've got one. how long term do we have data for? Because obviously, if we're going to like push the boundaries to Mars and things like that, we're going to be increasing the amount of time that we need to be able to train for. I guess bed rest studies have their limitation to be able to only last for so long. So what sort of duration of data are you working with to try and predict intervention and losses over such a sort of sustained and prolonged period of time?
Yeah, currently the longest mission duration on average is about a year.
So a chunk of time. And what's the exercise adherence like for astronauts? Do they do what they're told, or do you have data on that?
Yeah, it's not, here's time and you can do it. We have flight docs that monitor them, the ACERs that monitor them. They may have preference on exercise device, so they may prefer a cycle over a treadmill, but they all do the exercise countermeasures. They all understand the risk or that are involved if they don't exercise.
I guess to follow up then, that's still kind of a year's time. And do you or is there any anticipation that this might be an issue with much longer missions, such as to Mars, where the scientific load might not be as intense, and there might be a bit more boredom and kind of setting in?
Yeah, that's a great question, and currently those are unknowns right now.
Yeah, I guess the kind of the exercise adherence literature and that health behavior might kind of research.
Yeah,
might
and the other thing is like isolation inside a confined space, you know.
Mm-hmm.
But the crew and the flight docs fight for their time. That's like, you know, it's mental health for them. They want that time. Most of them, all of them want that time. They're so busy and so stressed up there. It's kind of the time to kind of relax and get some mental health, at least from what I'm told with from the flight docs.
Mm-hmm. Yeah, no, that's a great point. Today, any questions?
Yeah, thanks a lot, Stephen. Maybe just a question. We're talking a lot about exercise, but are there any like promising nutritional approaches that you see? I mean, as maybe as a personal view on that, that could go either in line with exercise or, you know, help in general with the prevention of deconditioning.
Yeah, we're looking at protein, looking at increasing protein. I know we've done bed rest studies. I mean, this is more pharmacological on testosterone. Testosterone has shown to minimize the reduction in muscle strength and muscle mass. But I think from a nutrition perspective, from what I heard from the nutrition lab, it's hard for astronauts to maintain their caloric intake. So I think obviously if they're energy deficient, they're expending more than they're intaking, and that's probably one of the main issues as well. But they just don't eat as much up there. Their hunger isn't as high.
Okay, that's due to less hunger essentially, or yeah.
Mm-hmm, yeah.
Okay.
I mean, they're forced to try to maintain their caloric intake.
And maybe just the final one from my side, is there a timeline now for some planned deep space explorations already established, or this is now currently unknown? Or
yeah, within the next 20 years, we're looking to go back to the moon and to Mars. 20, 30 years. Yeah.
Great. Thanks a lot, yeah, for the great talk.
Mm-hmm. Yeah.
Eric, can you give us an insight into kind of, I guess, how many people are involved in kind of at Johnson Space Center and elsewhere to really develop all of these things? And how does the team kind of work? When you say there's a nutritional team, there's obviously like an engineering team, there's kind of doctors, like how, if, for example, you wanted to test an intervention or something like that, like how does that entire team decide, okay, well, no, we do want the individuals to be doing sprint type exercises, you know, and that's going to go under prescription?
Yeah, we have or an organizational structure that has a research study, a whole umbrella of research studies that get implemented to flight, that are flight studies. And the crew obviously consent, and they don't have to participate in the research studies, right? So they obviously have to consent to any study that they want to participate in.
But I mean more kind of in terms of the ground team at Johnson, when you're designing, like if you have an idea that you want to, again, as an example, really prescribe and test kind of test high intensity training as an example, like how do you coordinate that with people like nutrition, with kind of psychologists, spaceflight, like how does that whole kind of internal approval process work?
Yeah, it has to be internally funded through NASA first. So there has to be a grant mechanism, and there are grant mechanisms to do that. And then, you just work with the different labs that you need a contribution to for a particular study. And then there's an organizational structure that organizes everything.
All right, great. Any final call for any questions before we wrap up?
I've got one, a bit more, very much more general, Eric, if you don't mind. I was just wondering, some people listening might be interested in space physiology and think, how on earth does anybody get into it? So I was wondering whether you could maybe finish on either a brief commentary of how you got on it, or any hints and tips would be fantastic.
Yeah, there are internships available for different areas. I can, give a plug right now. We're hiring a PhD level exercise physiologist right now. You'd have to go to the website. If those are interested, I can send you the link in my email. So I'm just going to move it to the first. But yeah, there's a website that you can go to look for jobs, and it's not only civil servants. We have different contractors that NASA hires, so you can be a contractor that can work for NASA related research projects.
All right, so again, Eric has his email there. If anyone does have interest, certainly feel free to follow up and talk with him more about possible careers in space. So I guess I want to thank both Eric and Val for really insightful talks into both kind of, in a sense, some of the opposite extremes, some of the really bench-spaced work on really understanding the impact of spaceflight, and then also kind of more big picture, how NASA is actually trying to minimize those impairments and specifically on exercise countermeasures. So thanks again to Val and Eric for excellent talks, and want to thank everyone for attending. In series 3, we're going to go with monthly seminars, and so the next one will be going the first Thursday of each month. So the next one planned is, again, obviously for early December, and it will be with Josh Foster. Will be leading it and discussing the Heat Shield project, which is a large kind of European funded project on really protecting human workers in a changing and getting hotter type of environment. So that's going to be in early December. So we will be, again, posting announcements on Twitter as we get closer to the date. And I look forward to seeing all of you there. So again, thank you for your attention, and hope you have a great rest of your Thursday today.